Okay. Hi, everyone. My name is Tait Algayer, if you haven't met me already. But this summer I was a summer fellow here, and today I'll be talking about some of my work from the fellowship that focused on tracking the dispersion of larval fish in the beautiful Lake Michigan. Can I use this? Or no, do I have to hit the space bar? Okay, cool. So a little bit about me. I grew up in New Jersey by the Jersey Shore. I went to the College of New Jersey for undergrad and majored in biological sciences. Did some work with Alaskan snow crabs there and wanted to continue my research. And so I started graduate school at Washington State University and I study evolutionary biology. And I work with migratory genomics in Pacific salmon for my thesis, and it's really cool. And I was really excited about this fellowship because I just thought it was really cool applied research project with genetics and work that I was already had experience in, but applying it to a new system and answering new questions. So Lake Michigan is home to invaluable living natural resources, which are the center of recreational fishing in the Midwest. Although most conservation efforts are centered around the top of the trophic system, changes at the bottom of the food web can drastically impact top level predators in the ecosystem. For example, the invasion of zebra and quagga mussels, collectively known as dreissenid mussels, have caused stark declines in populations of Chinook salmon and walleye in the Great Lakes. And the link between these organisms are two crucial intermediate species known as yellow perch and alewife. Those were kind of the focus of my project, and I'll be talking more about them. Alewife are an economically important prey fish in Lake Michigan. Initially an unwelcome invasive species in the Great Lakes, alewife became the primary food source for stock Pacific salmonids, supporting a multi-billion-dollar recreational fishery. And yellow perch are native to the Great Lakes and have long been the focus of fisheries managers. They're an exploited fish species themselves, but they also serve as a forage fish for walleye. So this is what alewife and yellow perch look like in their adult life stage, but since I'm interested in studying larval fish, I'm used to them looking like this. They're very teeny tiny, just a few millimeters long. And both species have undergone significant declines in Lake Michigan since the 1990s and then have experienced variable recruitment success since then. So a combination of upper and lower trophic effects have led to these declines, but one phenomena that has greatly impacted the recruitment success of both of these organisms has been the introduction of dreissenid veligers, which are the larval phase of dreissenid mussels. So the normal diet of larval alewife and yellow perch are zooplankton and rotifers, but I guess when you're a tiny little fish, it's easy to confuse these two things. And the dreissenid veligers are actually a lot easier for them to capture. So they eat a lot of those, but it's been detrimental to them because they lack a lot of nutritional value. So Ed calls it a fatal attraction. These larval fish have a fatal attraction to these dreissenid veligers. So that's been one cause of declines that you can see starting in like the 1990s when they were introduced. But since then, there has been a lot of variable recruitment success. And a large contributor to that has been changes in habitat, which are largely influenced by the advection of larval fish. Both alewife and yellow perch spawn in nearshore environments, making their larvae highly susceptible to advection via lake currents and lake circulation patterns. Since larval fish are so tiny and their swimming abilities aren't fully developed yet, they're largely at the fate of these lake currents, so they can easily get whisked away to either more favorable or less favorable environments for rearing. Currents known as coastal jets can undergo sudden reversals going from cyclonic to acyclonic due to changing wind directions or internal waves. Upwelling events, which are also largely unpredictable, are a main driver of larval fish advection as well. And ultimately, lakeshore currents greatly impact the recruitment success of larval fish and can be studied to learn if fish are being advected to more favorable or less favorable environments for rearing. Monitoring techniques for advection of larval fish includes physically tracking larval fish patches for by performing tows with ichthyoplankton nets. This is often a labor-intensive process, which I learned this summer, which involves a crew of scientists going out on a research vessel for days at a time to physically follow the movement of larval fish. And another way to track larval fish evection is through the use of larval dispersal forecasting. So many of you know about these satellite track drifter buoys that we have. You can release them into the water and then they float along with these spontaneous currents. And so through tracking those, you can predict where the larval fish might be. And then we have a numerical hydrodynamic model which predicts particle dispersion patterns. So using these kind of like mathematical modeling methods, we can better predict where the fish are going to be on the research cruises, so better informing our sampling methods. So, but still, this larval dispersal forecasting model has limited predictive accuracy and can still be improved using alternative methods, like eDNA. So luckily, eDNA sampling is a well-established method that can be used to monitor species in aquatic environments. eDNA sampling can reliably report species presence or absence through detecting short fragments of DNA that's present in the environment, and it's relatively inexpensive and accessible. It still has its limitations. Its ability to detect life history stage of these organisms or relative abundance are still underway. And eDNA can be present for lake environments for weeks or sometimes up to a month at a time, making it really hard to detect how recently that organism has actually been there. Also, sometimes dead organisms are present in that area and they can still shed DNA for a while. So that's called legacy DNA. And this has been a big barrier in assessing how recently species have been in the area. So legacy DNA can make it unclear how long ago the organism was actually present at a sample location. A new paper from Rao's group showcases the application of using eRNA monitoring to eliminate the possibility of detecting these legacy DNA signals. So this 2021 study performed temporal sampling with dreissenid mussels and found that ratios between eDNA and eRNA signals can be used to more accurately predict the age of this eDNA signal in the field environment. So since RNA is much less stable than DNA, it degrades much quicker. So that allows us to more accurately gauge when species were present at sampling sites. And since different organisms shed genetic material at different rates, we can't just like use our findings from this dreissenid mussel study. We wanted to perform laboratory studies with larval fish to see how fast they were shedding DNA and RNA and see how long their DNA and RNA signals were staying in the environment for. And then we also wanted to conduct a field study to see if using eDNA and eRNA were going to be a valuable method for informing sampling methods for tracking the dispersal of larval fish. So now I'll be talking about our laboratory study and what we did, and then I'll go on to talk about what we did on our field study. So the goal of our laboratory study was to look at rates of eDNA and eRNA degradation in larval yellow perch. We performed temporal sampling up to 17 days after the fish were removed from 5-gallon tanks. And we had tanks with 0, 3, and 100 fish present, and we did 3 reps of each. Then we filtered water at each of the time points. Down here, extracted DNA and RNA, and then ran QPCR to amplify our target gene, CO1, which is a mitochondrial gene that is largely conserved within the species. If you aren't familiar with QPCR methodology, I will give a brief overview. So we designed primers to amplify CO1. Those primers anneal to the CO1 gene sequence in yellow perch. This is specific to yellow perch. And a fluorescent dye binds to that sequence. 40 thermal cycles are performed to amplify the gene, and fluorescence signal increases with more synthesized DNA. And then you end up getting something that looks like this, showing either amplification or no amplification. And then you get cycle threshold values. Basically, the lower the cycle threshold value, the more amplification you are getting of that gene, indicating that there is that signal is fresher in the field environment. So here we have our degradation of DNA results from tanks with 100 fish. And you can see this blue line that it degrades exponentially after you're removing the fish from the tank. So here's 0 hours and then fish are removed from the tank, and that signal begins to like plummet as soon as you take them out. And there's kind of an inflection point of 24 hours, it really begins to drop. And after 48 hours, you're losing about 75% of that initial signal when we were there. So the main takeaway from this is that larval fish eDNA signals detected in the field are likely no more than 24 hours old, just because of how quickly they do degrade even in the laboratory environment. And we also saw some interesting results with our abundance data. So here, like I was saying before, here's the cycle threshold values. So the y-axis is reversed because the lower the signal, the lower the number, like the higher the signal. So we have results from 3 and 100 fish, and you can see that the degradation rates are like relatively the same, but the signal from 100 fish starts much higher than the signal from 3 fish. And this is pretty cool because we can tell that stronger eDNA signals can indicate greater abundance of larval fish. This has not been like a developed method in eDNA. Normally it can just detect presence or absence, but this is exciting because we have a way to quantify relative abundance in field environment with larval fish. Okay, on to our field study. The goal of our field study was to use traditional larval fish advection tracking methods to validify the reliability of our eDNA tracking method for alewife and yellow perch in the field environment. We went on 2 week-long research cruises on the Laurentian this summer, one at the end of June and one at the end of July. We did not have time to process our samples from the July cruise, so all the results that you're seeing are from the June cruise, which actually took place during a major upwelling event. So that's something to keep in mind when doing these results. We sampled 7 different sites in Lake Michigan over the course of a week. We just have our transect site here. Sites 2, 3, and 4 were based around the model predictions. Site 2 here is called the centroid. That's the site that's predicted to have the highest density of larval fish. And then we have sites 5, 6, and 7, which were based around the trajectory of the satellite track drifter buoys. So on day 1, we went here and we found fish, and we were like, put in the drifter buoys. And so we put them in, and then they floated over the course of 3 days, and they went down here. So we would predict that if there were larval fish there, that they would float along with the drifter buoys, and that we would find them again at the drifter buoys a few days later. And at each of these sites, we sampled water for eDNA and eRNA, and then we did our ichthyoplankton tows. We caught alewife larvae at 4 of the 7 sites we sampled and got eDNA signals at 6 of the 7 sites. Stars indicate sites where we caught fish, and the green indicates sites where we also got a positive DNA signal for our fish of interest. We did get stronger eDNA signals at sites where there were higher densities of alewife larvae, supporting our quantitative findings for our laboratory study. This is exciting, but we only did 7 sites on this cruise. We have 25 sites from our July cruise, so hopefully that will greater confirm that if we get a higher signal, that also means higher abundance presence. Another exciting thing is that we were able to detect eDNA at sites we were not able to catch larval alewife, indicating that eDNA is able to give us some insight that our current tracking methods are not able to provide us. So you can see sites 3 and 4, we did not catch alewife there, but we did get eDNA signals telling us that fish were probably there within the past 24 hours. We still do need to run our trials for eRNA just to make sure that this is to make super sure that this is not legacy DNA. From our trials with Dreissenid mussels, it was found that eRNA degrades within like 2 hours. So if you're getting that signal, like you bet that they were there, because eRNA just like degrades so rapidly. And for yellow perch, there was not a lot of them. We caught them at 2 of the sites that correlated with the trajectories of the drifter buoys, and we found them in much lower densities than the alewife. eDNA was not able to detect yellow perch at 1 of the sites where they were found. You can see right here, it's a star and it's also red, meaning that we did not get a signal. This was also the site with the lowest abundance, like for alewife and yellow perch. So this just means that eDNA may not be able to pick up on lower abundances of fishes. Some potential means for compensating for this could just be filtering more water. We only filtered 300 milliliters of water on the June cruise, but on the July cruise, we ended up filtering 1,000 milliliters. So we'll see if that gives us better signals for lower abundances. You could also perform more reps at each site. And on the molecular genetics side, you could also decrease the threshold for QPCR. So basically saying like, hey, if something is at a low level of expression, I still want it to be detected as like amplified. So you could also change that threshold on the molecular level as well in order to get a signal with lower abundances. Some future work includes running our trial for eRNA findings, since we didn't have time to do that within the fellowship period. And we also have to process samples from the July cruise, which did not take place during a major upwelling event. So it'll be interesting to compare the results from upwelling cruise versus non-upwelling cruise. And our group is also interested in sequencing the gut contents of larval fish that were caught on the trip to explore if alewife and yellow perch larvae are feeding upon Dreissenid veligers in places where there is high zooplankton abundance. So we've designed like the primers for all the zooplankton species and the Dreissenid veligers. We just like had really like high ambitions for this project, and like we were not able to do it all due to like the length of the fellowship period. But this is also just like a cool thing that's a part of this project. But we're just waiting on the gut contents to be extracted from these teeny tiny larval fish, which is obviously a very tedious project because they're so small. But yeah, that's another question of this project is, are they eating the veligers and is it influencing their abundance at the sites where we sampled? In summary, since early success of larval alewife and yellow perch is largely dictated by advection patterns of larvae, monitoring these patterns can help inform predictions for population health for valuable fish species in the Great Lakes. Back to our initial question of can eDNA enhance the monitoring of larval fish advection in the Great Lakes? We say yes. We were able to pick up eDNA signals at sites where we were not able to predict that they were there with the model, nor catch them with the ichthyoplankton nets. But we still need to perform our eRNA trials to ensure that this wasn't a legacy DNA signal and that they were really there within the past 24 hours. And although this method is promising, there were some limitations where we weren't able to detect them at lower densities, but I think that we could easily overcome these limitations by performing more replicates, filtering more water, or changing QPCR thresholds. Okay, big thank you to everyone here at Sigler. This fellowship has been so awesome. Thank you to my mentors, Rao and Ed Rutherford. You guys are great. I've loved working with you. And our research cruises out on the Laurentian were super fun. And yeah, thanks to Gloral and everyone here. I'll be happy to take questions.
Questions for Tait. And I should say too, if people have questions online, please either raise your hand and Aubrey will help you unmute, or you can just enter them in the question box. So questions for Tait.
How do you think the upwelling would impact the results?
Would impact the results of larval fish abundance or with eDNA? Both. I don't. I think maybe it could impact how long eDNA is like maybe hanging out for, like if there's more circulation happening during those times. As far as larval fish abundance, I mean, it creates a less favorable environment for larval fish in general. So I would expect there to be lower abundances of larval fish during those upwelling events, and probably for the eDNA signal to not hang around for as long.
Hey, Tate, that was great. Even though you didn't get to everything you planned, I'm amazed at how much you got done.
Thanks.
It was awesome. I just had a question about the gene marker, the CO1. I was wondering if you could say more about, why you chose that one, and if there are other alternatives that might change your sensitivity or maybe not resolution, but
Yeah.
comment broadly on that. Thanks.
So that's a good question. CO1 is a mitochondrial gene, and mitochondrial genomes are typically largely conserved within species, and they're also typically just like expressed pretty highly. So when we're designing markers for eDNA, we want them to be highly expressed. So that just means like the gene is like turned on at all times. And since we're designing our markers based on the sequence, we want that sequence to be highly conserved. So CO1 is pretty great for that. Also, when we're designing markers, it's better to use mitochondrial versus nuclear, just because when you think of a cell, there's one nucleus, but there's so many mitochondria. So it's easier to get that signal if you're designing it around a mitochondrial gene. For alternative ones, we did make alternative primers for ribosomal ones. We made it for H2B and 18S, but we didn't have time to run those trials. But those are alternative ones. Another hope is that we can look at life history specific primers. So that would be looking at genes that are differently expressed in different life history stages. So we would need to choose a gene that is not mitochondrial because those genes are pretty consistently expressed across life stages. So we have played around with looking at genes that are not mitochondrial in order to create life history specific primers. I hope that answered your question. Okay. Mm-hmm. Okay.
Great. Thank you so much, Tate.
Thank you.